My name is Dr. Marchetti Dario. I'm a director of the biomarker program at Houston Methodist in Houston, Texas. And the focus of our research is to capture, isolate and interrogate circulating tumor cells as seed of metastatic disease, notably to brain. And we are implicating these cells as a fundamental player in the metastatic process and possibly also in mechanism of tumor quiescence. And the focus of the research that I presented at Fondazione Menarini is particularly to identify a subset of these CTC which can be relevant in the progression of breast cancer to brain metastasis. And in a nutshell, we have keeping the presence or absence of brain metastasis as detectable by MRI, magnetic resonance imaging, as the standard of care. We have isolated CTC from blood from patient possessing either brain or no brain metastasis and interrogated these cells. In relation to discover, define signature of this CTC from patient not possessing brain metastasis versus one possessing brain metastatic disease. And the clinical implication of our work are twofold. First, in patients that have no brain metastasis, the translation of the idea is to identify these cells and signature which potentially allow us to identify brain micrometastatic disease or nascent brain metastasis that are not detectable by MRI. Therefore, it's a better, a more sensitive screening tool to define the onset of brain metastasis in this patient. Conversely, from the patient that are being diagnosed with brain metastatic disease, the clinical implication is to develop the research further and develop clinical novel CTC tests to be put in the clinical. One, to give the oncology a better treatment decision in relation to define biomarkers present in CTC subset. And two, also to define the efficacy of this treatment over time, therefore performing longitudinal study of this CTC, monitoring the decrease or the alteration or the modification of this biomarker expression over time and in the patient as a reflection of the efficacy of the treatment. And we consider this incredibly important because brain metastasis in general, extending from breast cancer, is an incredibly devastating disease. It is increasing in frequency, not just in breast cancer, but in all types of cancer. In fact, it is considered to be the #1 cancer killer within the next decade. Already in the United States, the number of brain metastases exceed the entire number of breast cancer diagnosed in that country. Second, there are no effective therapy. And basically the overall mean survival in these patients is dismal, only 2 to 3 months as a mean. And so therefore we consider these advances incredibly important to potentially to monitor and to combat brain metastatic disease and put forward as a background of this study to extend this study to other cancer types or organ types and target of metastasis. Therefore, using CTC as a clinical useful tool and translating into clinics, the concept of liquid biopsy. So I thank you for your attention, and I hope that you will be excited to look and listen to my lecture where we will present more details of this work that is being published in prestigious journals.